Hello students, this is Professor Fernetovich. I am finishing up our lecture on the fundamentals of chemistry, chemistry for botanists. We left off talking about cellulose as one of the polysaccharides in the carbohydrate group of macromolecules or polymers. And now we're going to talk about lipids. Again, this is part 4 of our 4-part series. So lipids are insoluble in water because they have long chains of nonpolar carbon-hydrogen units. And sometimes these are saturated, sometimes unsaturated. We have fats and oils. These are both called triglycerides, and they're for long-term energy storage. Fats are what we call that in animals, and oils in plants. Fats are saturated, oils are unsaturated, and in human use, we see fats as butter and lard in our kitchen and oils as cooking oils. There's also another type of lipid called a phospholipid, which is part of the plasma membrane. It's a lipid with a phosphate group attached to it, and that's a non-stick pan spray. We'll talk more about those as we talk about the cell because we'll focus on the plasma membrane and its importance. Those phospholipids play a real critical role in the function of the plasma membrane. There are steroids, types of lipids. They're on the membrane and they are hormones. Even in plants, we see those as medicines in human use. And plants produce waxes, which help plants to retain water and resist wear. Of course, in our human world, we use waxes for candles and polishes and that type of thing. So fats are saturated and oils are unsaturated, and the reason is shown here. It has to do with the molecular structure. So overall, these two are called triglycerides because they have 3 chains of fatty acids. 1, 2, 3. In a saturated fat, those chains are straight across. Those zigzag lines represent carbon and hydrogens. And all the carbons and hydrogens are bonded together in their maximum configuration. All the carbons are saturated with hydrogens, and that means that each carbon in this chain has 2 hydrogens attached to it. So this packs really tightly. It's straight across. It's like a block. And at room temperature, a molecule with this configuration is solid. Butter, solid. Oils, on the other hand, have a double bond in at least one section of a fatty acid chain. There could be multiple double bonds. We would call that a polyunsaturated fatty acid. And because of the double bond, the fatty acid chain bends. And that bending allows for more fluidity in this molecule. It doesn't pack as tightly, so it's looser, less dense, and we see that in our liquid oils. Really interesting chemical differentiation between saturated and unsaturated fats. Now, nutritionists are always arguing about how these affect our health, but I just want you to understand the molecular structure and how that affects the overall type of fat that we're looking at. Okay, again, we see saturated fats more commonly in animals and unsaturated more commonly in plants. So these are triglycerides, and this slide just takes you through what I just explained, written down. So if you want to take some notes on this slide, you can. One good thing to do on these PowerPoints, video lectures, is to print them out or just print out the PowerPoint which I provide and then take notes as we go. This is a saturated triglyceride, just shown here with all the carbons and hydrogens. See that? So here's our long chain fatty acid right here. Carbons. Carbon can have up to 4 bonds, so all of these carbons have 4 bonds and the maximum number of hydrogens that they can hold. This end one can have 3 because it has an end bond. And this is the glycerol part of the molecule. Okay, this is a phospholipid shown here, and this is one of the fatty acid chains here. here's another one. So a phospholipid is similar to a triglyceride, but it only has two fatty acid chains. These fatty acid chains are not the non-polar part of the molecule. Here's a double bond right here. You could see that. Very small, causes a bend here, so this is unsaturated. And the glycerol is right there in the head, we call that, of the molecule. So here's our glycerol, and here's the phosphate group attached. So this is where a third fatty acid chain would be attached if this was a triglyceride, but it's not. It's got a phosphate. Now that phosphate is polar, so that changes this molecule significantly. So it has a polar side to it, which has charge, and non-polar side. So this part is attracted to water and can dissolve in water, and this part repels water. Now that's really fascinating and important in the plasma membrane. And I know I said I was going to talk about this later when we talk about cells, but when this arranges in the plasma membrane, it arranges as a bilayer with the phosphate groups pointing towards the outside of the cell in this layer of phospholipids, and the phosphate groups pointing to the inside of the cell in this group of phospholipids. And then those nonpolar fatty acid tails are wiggling towards each other. So we get this kind of waxy nonpolar layer for the cell, which helps the cell to regulate moisture content. Okay, so these can interact with the watery cytosol, and these heads can interact with the aqueous extracellular environment. Okay, so we're going a little fast, but I want to talk about proteins. Proteins have many functions for cells. A lot of people think that they're just for muscle, but they function in support, all kinds of support. They're enzymes, which are so important for cells. Enzymes help reactions to happen inside a cell. They allow for transport across the membrane, defense, antibodies. Even in plants, they function in defense, and hormones. Plants have hormones, and we'll talk about those as we go. Auxin is one that's very important, a plant growth hormone. So proteins are polymers. I use the term polymer and macromolecule interchangeably. These are polymers of amino acid monomers. So amino acids make up the protein. And there are 20 different amino acids that are found commonly. And all of them have the same basic structure except for what happens at a placeholder. So this is a table showing the 20 amino acids I'm talking about. They all have a carbon, this carboxyl group, an amino group, and a hydrogen, and then something else, which is highlighted. And that something else is the R group that differs for each amino acid. And that's what defines the amino acid. So those are what really make each amino acid unique and help it in its function. So we put these together like letters in an alphabet constructing proteins. So a protein is a long chain of different a certain combination of amino acids. We've got tryptophan, leucine, isoleucine, and proteins can be over 100 amino acids long. So there's a lot of possibilities here. Now, the chain of amino acids is the primary protein structure. Okay, it's shown here as kind of like beads on a chain. And so each one of these would be an amino acid. They could be duplicates. We can have alanine, alanine, isoleucine, tryptophan, alanine. It all depends. Okay, people who study proteins map out what amino acids are in the row. Okay, this will then bend or fold, forming a secondary protein structure. This is called a beta-pleated sheet, and this is called an alpha helix. Then those will fold in on each other, making the tertiary protein structure. And then this will combine with other tertiaries to form the quaternary final protein structure. So forming a protein in a cell is a really big deal. It takes a lot of work for the cell, but that's what the cell is all about. If the cell had one major export or product, it would be proteins. Proteins are coded for by the DNA inside the nucleus of the cell. So what DNA, genetic material, does is tell the cell what proteins to make. And that brings us to our last macromolecule, the nucleic acids. Nucleic acids are polymers of nucleotide monomers, and we see DNA and RNA in the cell as examples. DNA is deoxyribonucleic acid. It's our genetic information in the nucleus. This forms chromosomes, and it's double-stranded in a helical spiral. RNA is related. It's a similar molecule, ribonucleic acid, and it's sometimes single-stranded, sometimes double-stranded. It varies in its appearance. And it functions in the assembly of proteins. So DNA tells RNA how to make the proteins, and ribosomes, which we'll learn about later, also help in transferring those amino acids into their order. So we see RNA in the nucleus and in the cytoplasm. Here's a couple pictures of DNA right here. Remember how I said hydrogen bonds were important in DNA? We see them more often in water, and we talk about that more commonly. But in genetics, we'll talk about the hydrogen bonds right here in the middle of the DNA molecule. It holds these two sides together. That's important. And this is a space-filling model showing all the different atoms of the DNA molecule. Here is a simple picture of RNA being single-stranded. It's like 1/2 of a DNA molecule. And what we have here are phosphates, sugars, and nitrogen-containing bases. And that's what we see in a nucleotide. Remember, nucleotides make up nucleic acids. I'll go back here too. We have the same type of thing, phosphates, sugars, and a base, nitrogen-rich base. That's what a nucleotide is. So I'm circling a nucleotide right now. The base, the sugar, and the phosphate right there. And that just repeats over and over again. The bases are different. We have C's, A's, U's, and G's. In DNA, we have those, except instead of a U, we have a T. So those are our 4 major groups of macromolecules, and that concludes our talk about basic chemistry for the cell. Now, we are going to keep going forward and look at how these molecules impact each other and affect the cell as we move into the next chapter, which is all about plant cells. So please read the chapter in your textbook, read chapter 2 in that Stearns Botany book, and check out Moodle for all the additional information. Thank you for paying attention, and please email me if you have any questions whatsoever.